All right, good afternoon, everyone. Thank you for joining me today. My name is Giselle Gonzalez, and I will be talking to you about the work I've been doing alongside Manuel Larac and Dr. Kevin Koo in synthesizing isoallantalactone, a naturally occurring product in Inula helenium. Now, this plant is a native to Eurasia, and it has been naturalized in parts of North America. The scientific name tells us that it belongs to the genus Inula, which has been known to have a variety of medicinal plants. The species is helenium, and we don't use the actual plant itself, more so the root of the plant has been used over hundreds of years to remedy a variety of health issues, such as alleviating pain, symptoms of the cold, and treating skin infections. Now, this can be due to the fact that Inula helenium has sesquiterpene lactones present in it. More specifically, it has isoallantalactone, allantalactone, and inulin. So as you can see right here, I'm pointing to the isoallantalactone, and it closely resembles its isomer, allantalactone. So it is believed that this functional group here with these oxygens is responsible for the medicinal properties of the plant. More specifically, we have antimicrobial, antifungal, and anti-cancer properties. And more recently, the spirolan furantones A through D, which you can see right here, that also closely resemble the isoallantalactone and its isomer, have been discovered. So these were isolated and extracted from the dry root itself, and they show very promising results in inhibiting the production of nitric oxide, which is important because it has implications in oncological, neurological, and cardiovascular diseases. However, it's a challenge to isolate and extract these specific spirolan furantones naturally from the plant, just because it is time consuming and we only get low yields. So our goal is to be able to synthesize these spirolan furantones. And in order to do that, we need to create its precursors of isoallantalactone and allantalactone. And now in order to synthesize the isoallantalactone, we require an intermediate known as 1,5-diketone. So in order to produce this intermediate, we use retrosynthesis, which just means you're working backwards, basically. So we take this product here, the diketone that we're looking for, and right where I have it highlighted, we create a break in the bonds. So this forms electrophile and nucleophile. This being the nucleophile, and this is the electrophile. Now, everybody in chemistry will tell you that this is what facilitates a reaction. You need a nucleophile and an electrophile. So further, we know that we can use silyl ether, which I have here. And now these arrows are just showing us basically the resonance, and resonance just tells us how the electrons move within the molecules. So we're able to obtain these electrophilic and nucleophilic sites that we're looking for. Now, in order to produce isoallantalactone, we used two approaches. So for the first one, as you can see here, we use the 2-acetylfuran. We brominate that. So we add 2 bromines here, and we have a yield of 75%, which is pretty good, given that we're usually shooting for 80% yield. We further synthesize this into silyl ether and react it with the methyl vinyl ketone to produce that 1,5-diketone that we were looking at in the previous slide, to give us a 35% yield, which means we could do better, right? So we're still working on that. We then perform a lithium halogen exchange to further remove one of those bromine and follow it up with a Wittig reaction. So now if this approach doesn't work, we have a backup approach. So for the second one here, again, we start off with the same starting material of the 2-acetylfuran, and we brominate that to get that brominated ketone that we saw in the previous slide as well. However, we take a different approach. We further protect this ketone group, and then we perform the lithium halogen exchange. So again, you see we remove one of these bromine groups, and then here we add a hydrogen to again reveal that ketone that we had protected previously. We react this with an enamine. This is known as a Stork-enamine synthesis, and further react this with the methyl vinyl ketone to again produce that intermediate that we were looking for, the 1,5-diketone. And again, we follow it up with a Wittig reaction. Now from here, both of these synthesis approaches follow the same steps to form the isoallantalactone. And as you can see here, we went from the Wittig reaction. So look at these 2 oxygen groups, and we remove them to create olefins. And then we attach this diester where the bromine was. And then again, we perform another Wittig reaction to remove this oxygen group and again, form an olefin. Now, down here are the steps that we haven't got to yet, but we perform this synthesis to create a cyclic circle right here. And we further create our isoallantalactone product. Now, my personal contributions to this project have been creating that 1,5-diketone with a silyl ether, which you can see we got a 35% yield. And then we've been doing a couple of experiments with the Stork enamine synthesis, which I have helped. And I also helped with the protection of the ketone group, which you can see has a pretty good yield of 95%. So in conclusion, we were able to synthesize this 1,5-diketone intermediate using the silyl ether. We're playing around with using the enamine, and we also did the ketone protection. Now, the future direction of this project could be to further synthesize isoallantalactone and allantalactone and use them to create those spiral amphiurentones that were recently discovered. Again, I have them here for you. And then we can further study these molecules to see how we can apply them to the future of medicine. And I would just like to recognize a couple of people for helping me along this journey of becoming a better scientist. The Q Lab, specifically Dr. Q, thank you for so much for allowing me to be a part of the lab. It was a very, very enjoyable learning experience. The RISE mentors, you all took a lot of time out of your day to help us out. Nee, thank you so much, and Dr. Brown as well. So I'm ready for any questions if I have any.